So I'm here today to tell you about how we target RNA polymerase I in the cell nucleolus. Those pink things are nucleoli, as a new approach to target triple-negative breast cancer. There's a huge unmet need for triple-negative breast cancer. It's one of the deadliest of all breast cancers. If you're diagnosed with metastatic disease, you have a 12% 5-year survival. It tends to occur in younger women. It occurs in women who are African American or who are Latina. There are no personalized therapies. So we've decided to take a new approach to target the cell nucleolus to discover new drugs that can target triple-negative breast cancer, or TNBC. This is me. I'm Susan Baserga. I'm a professor of biochemistry here at Yale. I've been studying the nucleolus for the last 30 years. I'm good at what I do. We've decided to take a new approach using our basic science expertise and turning it now to cancer therapy. This is not a new idea. We've known since the 19th century that the nucleolus drives cancer. This was the work of Giuseppe Pianese, who first observed that cancer cells had larger and more numerous nucleoli. And that brings us around to the idea that perhaps we could find small molecules that would then inhibit the most important enzymatic molecule in the production of ribosomes in the nucleolus, and that is RNA polymerase I, as you can see on the slide behind me. And in fact, we already target the nucleolus with some of the cancer drugs that are already in existence. They just weren't made to do that on purpose. And because they weren't made to do that on purpose, they have a lot of toxicities. So we think that we'll be able to use our new screening platform to find drugs specifically targeted for the nucleolus that would have less toxicity. Oops. So we've exploited a screening platform that my laboratory developed that is very simple. It just counts the number of nucleoli in a cell. Most cells have 2 to 3 nucleoli. If you disrupt ribosome biogenesis, it goes from 2 to 3 to 1, or 2 to 3 to 5 or more. We've used this screen in a number of ways. We screened genome-wide for siRNAs, also for microRNAs and long non-coding RNAs, and we've published many of these results, as you can see here on the slide. We've decided then to use this for small molecule discovery. And we screened this Life Chemicals FSP3 library, which is about 25,000 compounds. We have excellent screening statistics. We have about 200 active compounds, which is about a 0.8% hit rate. 137 of those were reproducible, and they can be classified into 11 different chemical structure clusters based on the Data Warrior software. We tested them to try to find a target, and we used an assay that was developed in my laboratory by 2 of my graduate students, Carson Bryan and Mason McCool. And it was an assay that assesses the activity against RNA polymerase I, this very important molecule in the cell nucleolus. As you can see on the slide, almost all of the compounds that we tested have activity against RNA polymerase I, shown here benchmarked against BMH21, which is a known inhibitor of RNA polymerase I out of Johns Hopkins. So we have a series of molecules that we now know the target for that we're then next going to test for their efficacy in killing triple-negative breast cancer cells in vitro. And that's what we've done here. And this is our most exciting data. This shows that the screen actives preferentially kill a triple-negative breast cancer cell line in vitro. We tested representative compounds from each of the 11 different chemical structure clusters. 10 of the 11 gave us differential killing. These are the best 3. So we just call them 1, 2, and 3. And as you can see, we compare the TNBC cell line, MDA MB468, against the near normal breast epithelial cell line, MCF10A. In 2 cases, we have differences that are as much as 2 logs, and in one case, for number 1, we have nanomolar efficiency. So we're very excited now to move these into mice to test to see if we can actually cure tumors. We have some competition. We have a group in Australia that's competing with us, and they're tumor agnostic, not dedicated to TNBC like we are. And they also target RNA polymerase I, as does BMH21 out of Johns Hopkins. But in both cases, these molecules were discovered by in vitro assays, not for the cell-based assay that we used. And we instead have a different approach, where we use a cell-based assay to discover molecules that are focused on TNBC. So I'm pitching for funds for proof of concept first in vivo tumor efficacy. All the work I've shown you so far has been done with funds from the Breast Cancer Alliance. The next step for these 3 screen actives is to test them at the Yale Center for Precision Cancer Medicine for the maximum tolerated dose in mice, and then to see if they can cure this TNBC tumor in vivo.
Well, that was perfect.
Maybe just based on the mechanism,
Yeah.
what do you think the potential toxicities would be?
So the toxicity you worry about is that it would affect normal cells just as much as cancer cells, but our in vitro data show, already that it's affecting cancer cells more. But I agree with you that we have to test it in vivo to be able to know for certain.